So in this section, I'm going to show you a part of the lesson I created for your lab and the Muscle PhysioEx. The link to this entire lesson is on D2L, but I'm going to show you part of the background information here just so it can help you visualize all of the processes that take place during muscle contraction. Okay, first we're going to review the steps of the muscle contraction, starting with the events that occur at the neuromuscular junction, as shown here in my little custom drawing. So I'm going to put one stage for each step on a separate slide so that you'll be able to pause in between steps so you can digest the information. But before we get started, I want to set up what the neuromuscular junction components are. So in this particular image, at the top here, we see the end of a neuron. So this would be a synaptic terminal, which is also called an axon terminal. And inside the terminal, there are vesicles that contain neurotransmitters. In the neuromuscular junction, the neurotransmitter is always going to be acetylcholine, which is abbreviated ACH. Also on the synaptic terminal are calcium channels, voltage-gated calcium channels, which are currently closed. And then down here at the bottom of the picture, we see the motor end plate, which is the part on the muscle cell that the neuron communicates with. So on the motor end plate, we see receptors that will bind to acetylcholine. So there are acetylcholine receptors shown here in orange, and they are also currently closed. The muscle cell itself, the plasma membrane, will actually dive down deep inside the muscle cell. These structures are called transverse tubules, or T-tubules. And then inside the muscle cell, you can also see the sarcoplasmic reticulum, shown in yellow down here. And inside the sarcoplasmic reticulum, you have a lot of calcium ions, which are the yellow dots. And the sarcoplasmic reticulum also has voltage-gated calcium channels, shown in green, which at rest are also closed. Down here at the very bottom is a small glimpse of a myofibril, which is where you find the sarcomeres. So I have a bigger picture of a sarcomere here just to go over some of its structures before we get started. So the dark blue lines are the myosin thick filaments. They have the little heads on them. The red lines are the actin thin filaments. For the sake of clarity, I am not including the titin proteins yet. And also be aware that this line, the two red lines, are called the Z lines, and these are the outer borders of a single sarcomere. And the M line is the inside middle line where the thick filaments attach. Also notice the zone of overlap, which is the area where the thick and thin filaments overlap. And then notice that this is a resting sarcomere, and all of the little heads on the myosin filaments are pointed away from the M line. The last thing to review before we get started is that in the space outside the neuron and the muscle cell, in other words, the extracellular space, the sodium ion concentration is high and the calcium ion concentration is high. And then inside the cells, so inside the neuron and inside the muscle cell, sodium ion concentrations are low and calcium ion concentrations are low, except for in the muscle cell where the calcium is inside the sarcoplasmic reticulum. But in the general cytoplasm area, the calcium concentrations are low. I wanted to go over just a couple of more terms before starting the steps of the muscle contraction. So let's zoom in to a portion of the sarcomere where we are looking at one of the actin filaments, shown in red here, and the myosin filament, shown in dark blue. You can see the little head sticking out. So what I wanted to go over is the fact that the actin filaments have active sites that are covered by a molecule called tropomyosin. And tropomyosin has a protein on it called troponin, which is a calcium binding protein. So it's important to know what these terms mean as we go through the steps. Now we will go through the 12 steps of muscle contraction, starting with step 1. An action potential travels down the axon of a motor neuron to arrive at the synaptic terminal. We will cover exactly what makes an action potential work in the next PhysioEx in the neuro section of lab. Step 2, the action potential causes the voltage-gated calcium channels on the synaptic terminal to open. And recall that calcium ions are high concentration outside the cell and low inside the cell. So when these channels open, calcium rushes into the synaptic terminal. Recall that at the end of step 2, calcium ions had come into the synaptic terminal. So with step 3, the calcium ions cause the vesicles, which contain the acetylcholine, to fuse with the plasma membrane, which releases the acetylcholine into the synaptic cleft, which is the small space between the synaptic terminal and the cell it is communicating with. So at the end of step 3, acetylcholine had been released into the synaptic cleft. So with step 4, the acetylcholine is going to bind to the acetylcholine receptors on the motor end plate, and these receptors are ligand-gated ion channels that allow sodium and potassium to pass through, but for ease of explanation, we're going to only focus on sodium. So these channels open, so think of these as being sodium channels. And then step 5, sodium ions are going to rush in, because remember, sodium is higher outside the cell and lower inside the cell. So sodium ions rush in, and this is going to trigger an action potential in the muscle cell along the plasma membrane of the muscle cell, which is called the sarcolemma. So at the end of step 5, we had generated an action potential on the sarcolemma, which is the muscle cell's plasma membrane. So step 6, this action potential is going to travel along the sarcolemma down into the T-tubules, where it will open the voltage-gated calcium channels on the sarcoplasmic reticulum, which will allow the calcium to be released from the sarcoplasmic reticulum and into the cytoplasm of the muscle cell, called the sarcoplasm. Now for the remaining steps, we're going to zoom into the sarcomere. So on the next slide, you're actually going to see a portion of the sarcomere. Okay, now we are looking at a zoomed in portion of the sarcomere. We went over the parts at the beginning of this lesson. And recall that at the end of step 6, we had calcium being released from the sarcoplasmic reticulum. So step 7, this calcium that was released in the previous step binds to troponin. And when the calcium binds to the troponin, that causes the tropomyosin to roll away from the active sites on actin. So now we have exposed active sites. So at the end of step 7, we had the active sites exposed on actin. So with step 8, the myosin head, which you should note at this point is bound to an ADP molecule, will bind with the active site on actin that is exposed, and this produces a structure called a cross bridge, which is where the myosin filament is actually attached to the actin filament. Also make a note that when the myosin head is bound to ADP, it really is attracted to the active sites on actin and wants to bind, whereas if the myosin head is attached to ATP, it does not like the active site on actin and does not want to bind. That becomes important in a moment. Okay, recall that at the end of step 8, we had the formation of a cross bridge. So with step 9, the myosin head releases the ADP and also a phosphate, which is not shown in this image, and this causes the head to pivot towards the M line. And because it's attached to the actin filament is also pulled inwards toward the M line. At the end of step 9, the myosin head pivoted toward the M line, dragging the actin filament with it. With step 10, ATP comes in and binds to the myosin head. And remember, when myosin is bound to ATP, it no longer likes to bind to actin. And so when ATP binds, this causes the myosin head to let go of the actin active site. Okay, myosin bound to the ATP has just let go of actin. And so with step 11, the myosin head is going to split the ATP into an ADP and a phosphate group. And the release of the energy by breaking down ATP is going to allow it to recock or reset into the original position, which was facing away from the M line. So we left off with the myosin head set back to the original position and attached to ADP. So step 12 is that the myosin will bind with a second actin, and the cycle will repeat over and over again as long as the tropomyosin is not in the way of the actin active sites, which requires that calcium still be around and bound to troponin. So as long as you have the calcium around, and as long as the tropomyosin is rolled off of the active sites, you will basically repeat steps 8 through 12, which will cause the actin filaments to be pulled further and further toward the M line, which would result in muscle contraction. So to reiterate, as long as the calcium is bound to troponin and tropomyosin is rolled away from the active sites on actin, the myosin heads will keep reaching up, grabbing a hold of actin, pivoting toward the M line, and sliding the actin filaments closer and closer to the M line. So as you can see in this illustration, as the myosin heads are pulling on the actin filaments are sliding into and close to the M line, which makes the overall sarcomere become shorter. And when all of the thousands of millions of sarcomeres in a muscle fiber are doing this at the same time, the muscle fiber becomes shorter. And when all of the muscle fibers in your muscle are shortening, the overall muscle shortens. And this is how muscles contract. They shorten, and then that causes them to exert a pull or a force on whatever they are attached to at their insertion point. So how does a muscle contraction end or terminate? First, we have to get rid of the calcium that is bound to the troponin. So the sarcoplasmic reticulum, which is illustrated up here in the left corner, has calcium pumps. And just as soon as the calcium is released, as we saw back in step 6, these pumps start working to bring the calcium back inside the sarcoplasmic reticulum. Remember from our first PhysioEx that a pump is moving a solute from an area of low solute concentration to an area of high solute concentration. So the pumps are taking the calcium from the sarcoplasm and moving it back into the SR. So with the calcium no longer around to bind to the troponin, the tropomyosin will roll back over and cover the active sites on the actin. And then once the active sites are covered, the myosin heads can no longer bind, and so the contraction cycle stops. So one of the stages of termination is that the removal of calcium causes the actin active sites to be covered again, resulting in the inability for cross bridges to form. Also, if you recall from steps 4 and 5, acetylcholine that was released by the synaptic terminal caused sodium channels to open, which allowed sodium to come into the muscle cell and generate an action potential. And as long as action potentials are being generated, calcium is going to keep being released from the SR. So we also need to turn off the synaptic transmission, and to do that, we have to get rid of the acetylcholine that is in the synaptic cleft. So there is an enzyme called acetylcholinesterase, which is abbreviated ACHE, and acetylcholinesterase breaks down acetylcholine. So in the synaptic cleft, this enzyme will break down all of the acetylcholine molecules, and as soon as the acetylcholine molecules are gone, then the ligand-gated channels close and no more sodium can come into the muscle cell. So the second event that causes the termination of muscle contraction is that the acetylcholine in the synaptic cleft is broken down by acetylcholinesterase and thus closing the sodium channels. So that is as far as I'm going to go here. If you want to take the quizzes and look at the rest of the information I provided for the lab, then this link is in D2L Chapter 10 folder under Lab Help, and you can go and do the entire lesson yourself.